This is a production of Cornell University.
So today I'm going to focus on one particular problem that we're working on that has nothing to do with seaweeds. It's plants, which is useful, I think, for this audience. And it's talking about anisotropic growth. And what you'll see in particular is that we're focusing on the hypocotyl as a system to study this, but obviously there's lots of places within a plant where anisotropy will occur. So extending these principles beyond that will be something that we're interested in doing next. So let's see if I can figure out how to use the pointer in this. I'm just going to use this. Okay, so our general question in the lab is trying to ask how shapes get grown in walled organisms. And I say plants and algae because I don't really look at yeast or bacteria. So I'm looking at multicellular walled organisms. And it presents a really interesting physical challenge. So you have a wall that's surrounding each cell, but also for the most part connecting cells to each other and connecting tissues to each other. And so physically speaking, when you want to have coordinated growth that gives you a shape or a pattern, you need to have coordination of that physical process of growth. And in general, as you'll see, I consider that as something that's really mediated by the cell wall. So here's an example of some of the systems that we look at and some of the organisms we'll use. But as I mentioned, we're really going to focus on, maybe it just doesn't have a battery. That's okay, I can dance around. We're going to focus on the hypocotyl today. So let's see. So when you want to understand how shapes are grown, oh, thank you. Whether you're talking about a cell shape or the shape of a tissue or a whole organ, you need to do different things. So first of all, you need to be able to quantify shapes. So we do quite a bit of differential geometry to look at how shapes change in time and how you can quantitatively describe them. You're going to talk about how you actually think that change in shape comes about. And to do that, we look at some physical principles and also some material science. So the cell wall is a material. And then we're going to look at how the cell wall biochemistry relates to those material changes and how the cell biology underlies that change in the material. And finally, of course, I'm a developmental biologist at heart, so I'm really interested in how the signals from the genetic program get translated into this physical change in shape. So today what I'm going to focus on when I start learning how to use the computer is mostly these two here. So what kind of cell wall changes are happening during anisotropic growth and what we think maybe some of the regulatory mechanisms for that might be. But there'll be a little bit of material science at the end. So this is my diagram of the cell wall. Just don't freak out. So it's an incredibly stylized, really simplified description of what we think a cell wall looks like. And pointing out that, of course, as some people in the audience will know, we don't really know what the cell wall looks like. So we know there are cellulose fibers in there. We know that that's embedded somehow within a pectinaceous matrix. And pectin is the chemical I'm going to talk most about. But there's also the cross-links, hemicelluloses that can happen in between fibers. And we're starting to learn a little bit more about the architecture of the wall, but it's an incredibly difficult structural thing to work with. And so a lot of the things that I'm going to say about the mechanics of the cell wall, we're taking a lot of assumptions into the structure as well. But hopefully we'll learn more about the structure and link that to the mechanics over time. But with a big grain of salt, we think that the cell wall has these strong, rigid fibers whose orientation can affect the material properties. But you also have the matrix within which they're embedded, which is what I'll focus on today. So our questions about the mechanical cell wall is, how does the wall change during growth and shape formation? What component of the wall or components might be important for cell growth or shape growth, and how they actually mediate that? And actually also how we can test the properties of the wall in vivo. And so as Adrian mentioned, I've been involved a lot in using atomic force microscopy as a way to test the material properties of the cell wall. And I'll go into that a little bit for you. So today what I'm going to do is focus on the hypocotyl. We're going to look at a role for pectin methylation in hypocotyl anisotropy. Then we're going to look at how maybe modulating anisotropy could have some interesting functional consequences for a plant. So instead of just saying, oh, that's a cool microscopy picture, why it might actually be useful to change your anisotropy. And then at the very end, if we have time, I'll do a little bit of what we're looking at to actually link the biochemistry of pectin to the material properties of gels and how we can translate that back into a plant. So it's probably a little bit superfluous for this audience, but the hypocotyl is an important organ. It's located between the root meristem and the shoot meristem in a lot of dicotyledonous plants. And what's going to happen is you need this extreme anisotropic elongation of this organ to get the apex out of the soil so you can start getting photosynthetic capacity happening and producing leaves. And so, where am I? Yeah. So this happens from the point of germination. You start getting this massive anisotropic elongation of this organ. And so that's the study system we're going to work with today. And obviously it's important for the early success of plants as crops. You know, the first thing that happens in their life is germination, but after that, you've got to get out of the soil. So one of the ways that we watch this happening is by simulating a continuous reach for the surface. So we grow seedlings in the dark. These are Arabidopsis hypocotyls growing in the dark and visualized using infrared light. And so what we can do is essentially make it so they never think they get out, and they're just going to continue this elongation until they run out of resources. And so what we can do from that is we can monitor length over time in really precise ways, so we can get quantitative descriptions of this elongation as it happens. So they start doing some fun things at the end, which I'm not going to talk about, but I like these wiggles as well. Start dancing. So as far as anisotropic growth goes, that elongation more in one direction than another, we have both a direction for anisotropy, but also a magnitude. So in this case, it's pretty easy. The direction of the hypocotyl is pretty much always up. So it's not that it goes up and then it starts getting fatter. It's a continual direction for the anisotropy. But the magnitude actually changes in time. So what I'm going to show you in the next slide is that you start off relatively slow, but then it gets faster. And that magnitude has to do with differential regulation of the elongation growth and the width growth of cells, which I'll also show you some data for. So if we take our movies and we quantify the length of the hypocotyl over time, and you've probably seen this before because this is a well-known phenomenon, you end up with what some people call a sigmoidal growth curve. So it's sort of slow increase in length at the beginning, and then you get a transition around 24 hours after germination to this more rapid elongation. And so that's what we're sort of tunneling down into. One of the things is what's controlling this transition to the rapid elongation, but also underlying that is what's making sure that most of the growth is going into length and not a lot into width, which would be a waste of energy if your job is to get you up out of the soil. Okay. So this really is a change in growth magnitude, anisotropy magnitude. So what we did is we looked at the cellular level as well. So we wanted to know what cells were doing at this point. So this has been done once before, and it was with scanning electron microscopy, and they really just looked at cell length over time. But if you think about it, cell length and width, that ratio is what's important also for anisotropy. And if you only ever measure length, you may be losing some information by not looking at width. So what's happening in this colorful slide is mostly the elongation of the hypocotyl happens by cell elongation. There's not a lot of division, at least in the epidermis. And so that allows us to track or pseudo-track cells by position. So you have 1 to 17 cells per file. And because these are growing in the dark, as soon as you take them out to image them, they stop growing. So we're pseudo time series by sampling at each time. And so then what we can do is we can say, well, this is cell 1, 2, 3, 4, and we can calculate individual growth curves and individual growth rates by position along that file. Does that make sense? So the first thing that you'll see is if you just look at the top of this, there's that transition to fast elongation. And what you may know already, which is known in the literature, is that all the cells don't start elongating at the same time. Instead, you get this, what we call an acropetal wave of elongation. So the cells at the base start elongating before the cells up near the top. And that happens in a wave. You can see it really nicely here. These are still small cells in length here, whereas these are elongating. And that wave of elongation actually travels up the length of the hypocotyl. So you have what looks like a pretty simple behavior, slow growth, fast growth for the whole organ, but it's underlaid by this interesting wave of elongation that moves from the base to the top. And that allows us to look at elongating and non-elongating cells, or fast and slowly elongating cells at the same time. So what's over here is sort of the first real measurements of width that are happening at the same time. And what you can see is that the change in width doesn't show that acropetal wave. And the cells are expanding in width very slowly. So this is a full order of magnitude different scale. So you're expanding in width really slowly compared to length. And you're not having that position-dependent control of where that growth in width is happening. So you don't get that sharp transition to a fast growth either. So this really is indicating that the growth in width and the growth in length are somehow being independently regulated. That's what we might say. Okay, this is the cellular level mechanism by which that organ level behavior is coming up. But what we wanted to ask was how that transition to rapid growth was achieved. And what that really means is what's happening at the base where those cells are elongated versus the top where they haven't started quickly elongating yet. And then also how that direction going more up than in width is actually happening. And so there's obvious roles here for cellulose orientation, which everyone will know about. And I'm going to hope to add to that a little bit today with a pectin story. So let's start with this. How do you get that transition in rapid growth? So if we go back to this data, we're really interested in that sharp transition where things start getting really fast, and it's because the cells at the base are starting to elongate much more quickly. And that allows us to ask this question about the cell wall chemistry between the fast and slowly elongating cells at one point in time. And so because of some work that we'd done previously in phyllotaxis looking at pectin chemistry, we knew that there was some link between pectin and where you would get a new organ forming at a shoot apex in Arabidopsis. And so we wanted to ask whether or not we could see similar changes in pectin chemistry here along the hypocotyl due to that expansion gradient. So I'm going to introduce Homogalacturonan to you. This will be the most biochemical the slide gets. So there are people in this room whose biochemistry is better than mine. So I apologize. But basically in Arabidopsis, the major cell wall pectin is Homogalacturonan. So it's a really simple chain of galacturonic acid. And when it comes out into the cell wall, it's esterified. So these little orange balls are meant to be those methyl ester groups. And what'll happen once you're sort of in the cell wall is that you can be selectively deesterified by Pac-Man or pectin methylesterase. This then takes off some of these methyl groups. And then what can happen there is that you can then cross-link with calcium. So this is this really canonical calcium gelation that'll happen in pectin gels. It's analogous to what happens in alginate, although the chemistry is different. But you get this calcium cross-linking, you get rigidification of a gel. So what I've got in this table is sort of what we predict the mechanical character of gels made of these different types of pectins should be. So when you can't calcium cross-link, you should be generally more soft as a material. As soon as you can calcium cross-link, you should be more rigid. That makes sense. It's like making jam. You're setting the jam. And we have some antibodies that actually let us look at the presence of these different epitopes within the cell wall, which is what we're going to look at next. But we can make some hypotheses about what we think might be happening with respect to growth. So when the pectin is more methylated, it should be more soft, maybe even more fluid. Growth should be easier in that case. The cell wall material should deform more. Versus when you have calcium cross-linking, things are a little bit tougher, and it might be harder for the cell wall to actually extend. Now, the reason that this one in the center says it's possibly rigid is because there are more complicated dynamics that can happen. You can have demethylated pectin degraded by polygalacturonase. And so if you're chopping your pectin up, that's definitely going to be a softer gel than if it's just, sitting there without calcium cross-linking, because you're actually chopping up the pectin backbone. So just because you have PME activity doesn't mean you're always going to be more rigid. It depends on presence of polygalacturonases and also presence of calcium ions. Okay, let's take a look at what these antibodies look like in that elongating hypocotyl. I think, oh, yes, I have to mention that we also have some proteins that we can use called pectin methylesterase inhibitors that basically stop this enzyme from deesterifying the pectin. So we predict that they should keep the pectin in a more fluid state. Whereas if you overexpressed PMEs, you might end up with pectin in a more rigid state. And then we'll come back to that in a minute. Okay, so these are transverse sections of hypocotyls at 24 hours post-germination, when we know the basal cells are actively quickly elongating and these top cells are slowly elongating. And so LM20, this is the methylated pectin. Hopefully what you can see here is that there's more signal down at the base than there is at the top, although the quantifications with these things can be very difficult, so I caution you. But up in the slow-growing cells, we definitely have more labeling with the 2 rigid or possibly rigid antibodies for Hg. So what that means, if you look down at the base, is that you should have more of the fluid pectin, and up at the top, you should have more of the rigid pectin. And so the idea is that the cells at the base are able to elongate, or part of the reason they're able to elongate is because you're keeping that pectin in a more fluid state. So it's getting delivered and it's not getting converted into the more rigid able pectin. Okay, so if that's true, can we actually test that? And this is where the atomic force microscopy comes in. So hopefully, I'll start this movie while I talk. So this is an image of an AFM experiment going on. And what you have is a very small tip here, which is actually going to do tiny indentations along the surface of the hypocotyl. So we're limited to looking at the epidermis right now, but we can poke. And essentially what you're doing is asking, if I apply a force, for example, to the surface of this table, if I apply the same force to the table, how much does it indent the table versus how much that force would indent my arm? And so by looking at the force and deformation that you get at different areas along a hypocotyl or between 2 different materials like me and the table, you can learn something about their material properties. And if you do this quickly, you're going to be looking at the elasticity of the cell wall. So if we do that at the base of the hypocotyl, at 4 hours, so when everybody's growing really slowly, this is the transition to rapid growth at the base, and this is when things are really kicking it. What we can see is that this number we get called the indentation modulus is going down in time. And so an indentation modulus just relates the force to the deformation that you get. And so the higher it is, the more force you need to apply to get the same deformation, and the lower it is, the less force. So it implies that the material is getting more elastic over time, which correlates really well with this increase in growth. Okay, that makes sense. This is nice and consistent. But what happens if we alter pectin chemistry? So based on our predictions of what's soft and what's allowing growth to happen, if we mess with the Homogalacturonan structure, we mess with its mechanics, maybe we'll also change growth. And so we're going to go back to those 2 enzymes, the PME and the PMEI, the inhibitor, to try and change the kind of pectin that's in the cell wall and see what happens to growth. So as a little reminder for you, so PME is catalyzing the removal of methyl groups. So this should allow pectin, if there's not a lot of polygalacturonase present, to calcium cross-link and become more rigid. But if you have inducible PMEI, that should block PME's activity and leave you more in this fluid state. And so we've done this with alcohol-inducible PME and PMEI lines, but there are only 1 PME and 1 PMEI, and there's about 60 to 70 of each family in Arabidopsis. So there are a lot of these genes out there. But in this particular case, we're just taking 1 PME and 1 PMEI and asking what happens if we inducibly overexpress them to the growth of the hypocotyl. So luckily, I don't know, this is just what happened. So what you can see here in gray is that normal, non-transgenic, but treated with the inducer, which in this case is ethanol, we get the slow growth and then the transition to rapid growth. And what happens when you overexpress the PME is that you kind of just always have slow growth and you never get that rapid transition. Probably, and we've done the biochemistry to look at this, because you're only having conversion of all the pectin into that calcium cross-linked form, or pretty much all of it. So everything's really rigid, everything is slowing down. And the opposite is true when you have the PMEI, which keeps all the pectin that comes out in that fluid state, and you're growing much more quickly earlier, and you sort of have a more flat, fast growth throughout the growth of the hypocotyl. So it has the predicted changes we would expect on an organ level, but also a cell level. So because of the way the induction works, we don't see the phenotype right away. You see it sort of at 48 hours, we can see a little bit better. So this is cell length. They look the same at the time of induction, but 24 hours later, you can see that the wave of elongation is happening. So this is the cell index from the base to the top. The PMEI cells are a lot longer at the base versus the PME cells, which are pretty short all the way along. So we're having that change in the organ and the change in the cells predictive based on the chemistry. So that's pretty nice. I will note that we still don't know why changing pectin elasticity actually has that effect on growth. So that's something we're working on. What is the actual physical thing that's happening in the cell wall that says, because your gel is rigid, you can grow less? So maybe next time. Essentially what happens is that PME blocks this rapid growth phase and PMEI is promoting it. So what we would want to see is that we can see the change in mechanics that we also expect to happen. And so luckily that also works out with the AFM. So now I'm showing you the AFM data in a map, first of all. So each point along this is a single indentation experiment. So you can get some spatial information as well. So when you have PME expression, things are generally more rigid. This is that indentation modulus. So the higher this is, the more rigid the cell walls are. And here we have PMEI, and things are a lot softer, more elastic. I'll note that these experiments are done in plasmolytic conditions because the cell wall is quite thin compared to the turgor pressure inside. When you're doing these indentation experiments, depending on how you do them, oftentimes you need to reduce the turgor pressure so you're not just measuring pressure when you're poking on a cell. And so I'll show you a graph now, so it's a little bit more quantitative, but you can see that the indentation modulus of the non-transgenic is almost halfway between when you induce PME and when you induce PMEI. So they're having that mechanical effect and the effect on growth we'd expect. Okay, this all sounds pretty good. But now I want to ask this question about how the direction in anisotropy is achieved. And what you might have noticed if I go back one is one of the things you get is some spatial information, and you might notice that these transverse walls appear to be more rigid in the non-transgenic than these elongating walls. And a really easy way to explain that is because these walls are just thinner. So if you push on something that's thinner, it will feel softer, regardless of if the material is different. But we know we've actually done some measurements on cell wall thickness here, and we know that the walls are maintaining the same type of thickness. So we think it's actually due to this chemical difference in the pectin. So those walls appear to be more rigid. The slow walls that might control width expansion are more rigid than the elongating axial walls. So let's investigate that a little bit more when we ask how that direction in anisotropy is achieved. So we looked at 24 hours, which is close to what you saw before. We were looking at 48 with the induction, but this is 24, and already we can see these walls are more rigid than the fast elongating axial walls. But even at 4 hours post-termination, when everything's growing really slowly, we can still see that there's a difference already in these transverse versus axial walls. So this is something that's set up pretty early and is coincident with the fact that that growth is always anisotropic. You're always elongating more in length than you are in width. It's just that it gets faster past 24 hours. And so we've done some quantification of this as well, which is maybe a little bit more believable. So at 4 hours, we can see that there is a difference between the transverse and axial walls. The transverse walls are more stiff. You see everything gets a bit softer by 24 hours, which is what I showed you before, but you can still see that this maintenance of the ratio between slow and fast growing walls is maintained. And so you may also have noticed that pectin could be involved in this as well, because in the PME and PMEI overexpressors, which we'll look at again, we've actually abolished that difference. And so now we're looking at transverse sections, longitudinal sections of hypocotyls. And we're looking at hypocotyl cells near the base at 4 hours, and then we're going to look at 24. But remember, even at 4 hours, we could see this difference in the elasticity of the cell walls. And so what I have marked here with red asterisks are endosperm sections. So, and then this is epidermal files marked in green, because it's kind of hard to orient yourself. So what you can hopefully see is that in the epidermal file, these two more rigid pectin markers have higher signal on the transverse walls than they tend to have on the axial walls. And the opposite is true for the softer pectin marker, LM20, where you have more signal on, on the axial walls and less on the transverse. Again, these are really tricky to do quantitatively, but it jives pretty well with the mechanical measurements that we can make. And we can look at this at 24 hours as well and see once these cells are starting to elongate, we're again seeing a little bit more of the rigid markers on these transverse walls here. more of the soft pectin on the elongating walls. You can see it gets a little bit more complicated. There is some rigid pectin on that elongating wall as well. So it's not as simple as saying, well, if you have this pectin, you will grow, and if you have this pectin, you won't. But it's really difficult right now to work out that quantitative relationship. So we're working on it, but it's pretty difficult, especially with these techniques. Okay, so you have differences in elasticity between slow and fast-growing walls. That same difference is mirrored between the slow and fast-growing regions of that acropetal wave. And we can correlate really nicely the chemistry of the pectin with the elasticity of the walls and growth. So when we go back to those PME and PMEI overexpressors, so this is the same image that you saw before, but now we're focusing on the fact that the ratio between the transverse and axial walls has actually disappeared. So the non-transgenic treated with ethanol shows the rate difference, but the PME, everything is rigid, there's no difference anymore, and the PMEI, everything is soft, there's no difference anymore. And you can see that before I showed you the length of the cells, but now I'm actually showing you the length versus the width, and you can see the PMEI cells are way more anisotropic. So the ratio of length to width is more, and the PME cells are less anisotropic. So you're affecting the anisotropy as well as the elongation rate. So one question that came up in the literature based on this observation is whether or not elastic asymmetry is actually enough on its own to drive anisotropic growth in the hypocotyl. So if you just pretend that cellulose anisotropy doesn't exist, if you just have stiffer cross walls than you do axial walls, is that enough to give you anisotropic growth? So we've done a little bit of computational modeling with Henrik Jansson's group at the Sainsbury Lab in Cambridge to ask this question. And again, all the assumptions that go into your model change perhaps what your outcomes will be, but it looks like in our hands, if you take this, we're doing this with a finite element method model, so we can prescribe material properties to the different cell walls. We can say, you should be more elastic or less elastic than these other direction walls, and ask then if we simulate growth, what happens? And it's using a modified Lockhart model. So what happens, we measured about a twofold difference between the transverse and axial walls and elasticity. If we add that in, the thing just balloons. So that twofold difference in elasticity is not enough. And maybe, we're not getting the true measurement, who knows? But if you pump it up to 100-fold difference, it is enough to give you anisotropy. So what you're looking at for the colors here are how anisotropic the strain or growth is. And in this case, the direction of the anisotropy is shown here with the white lines. So with nothing, just everybody's all the same, you end up growing more in width. But if you do this 100-fold difference in elasticity between these walls and these walls, then you can get some anisotropic growth. But we think that's probably pretty unrealistic. And it's pretty unrealistic to believe that pectin would be the only thing in controlling anisotropy, especially since we know that cellulose is important. So what we did next is we simulated some anisotropy from cellulose. And in the hypocotyl, it's not straightforward in the way that it is in Paul Green's original work in Nutella. So in those instances, you have cellulose fibers wrapping around the girth of the cell, and that is meant to restrict expansion in width. But if you can allow those fibers to slip apart, you can facilitate growth in length. And that's this canonical way of controlling anisotropy. In some organs, that tends to be true, and in some species, that tends to be true. But remember, we're looking at the epidermis here, and in Arabidopsis, the epidermis doesn't show really strong alignment of cellulose. What we know now is that the cortex shows pretty strong alignment. So what we've done in these simulations is not say that the epidermis itself has to have that material cellulose anisotropy. We're just providing it either from the interface of these cells, or actually what you'll see next is from inside layers of tissue. So no matter where that information comes from, what happens is that you get anisotropic growth when you have cellulose anisotropy. That's good. But if you add on this twofold difference in wall elasticity, you actually get more anisotropic growth. It's stronger anisotropy and it's faster. So you can sort of see that here. This is with the twofold elastic asymmetry added. This is before growth. And after growth, you can see the cylinder's a little bit longer. And the degree of anisotropy is a little bit higher. And it's even more profound if you simulate internal anisotropy, but again, these numbers are kind of just made up. But again, you see that additional thing. And so what we think is that this pectin asymmetry in the epidermis, it's not the only thing controlling anisotropy, but it's helping anisotropy to happen. It's helping that growth to happen a little bit faster. And it's making sure that those walls are elongating a little bit faster than these transverse walls. And that could be a really important developmental advantage. Okay, so obviously we want to know what regulates that anisotropy, and I don't have an answer for you today, but we're looking at some cytoskeletal dynamics and delivery and endocytosis and exocytosis balance, but also some of these other modifying enzymes. Where do they get delivered? Where are they activated? But we might also be interested in whether or not the regulation of anisotropy can actually be functional. So why would you want to get out of the ground faster? I mean, it seems to make a lot of sense, but let's see. Okay, so let's go to the second point, which is looking at modulation of anisotropy and growth plasticity. So this little guy has to elongate out of the soil. It makes sense you might want to do that quickly, right? Because then you can photosynthesize sooner. You establish yourself, maybe you beat some neighbors out of you shade competition, you get out faster, you establish yourself, so everything's better for you. But there's a lot of environmental variability that they're dealing with. So what kind of soil do they have to go through? Does the compaction of the soil change whether you'd want to go thin and fast or fat and slow? How much carbon and other nutrients are available to you? And maybe you want to respond to that in a different way. And so there's all these complicating factors that go into saying, well, maybe being faster isn't always better. And so what we noticed in the lab is that if you grow a Arabidopsis, this is very, very natural. If you grow a Arabidopsis on plates with sucrose, their anisotropy is actually different. So what it looks like in this video is that these are shorter. They're slower and they're shorter, and that's kind of, you would think, well, maybe that's a bad idea. But when we calculated their volume and their volumetric growth, what we saw is that it was identical. So they're growing the same amount, they're just patricianing that, my English, I don't have an excuse, but they're giving less growth in length and a little bit more in width. So the volume maintains, but their partitioning, I just stopped saying that word, is different in length and width. So when you have sucrose, you're less anisotropic. Okay. So I can tell you that this is not based on ethylene, which is a possibility. So you'll know that the triple response involves you getting really short and really fat. But if we do this in the ACS ethylene synthesis octuple mutant, you still get this response. So it's not dependent on there being ethylene present, which is a good control. And it also happens no matter what kind of sugar you throw on it. So we're looking at the anisotropy index. So this will be the ratio of length to width. And you can see normally they're way longer than they are wide. When you put them on sucrose, that drops because they're getting fatter. And it happens if you have sucrose, glucose, dextrose, fructose, galactose, trehalose. So it doesn't appear to be specific to any one carbon source. And the other interesting thing is that if you chop off the cotyledons, which are an endogenous source of carbon, you mimic the low sucrose condition. And if you add little grains of sand back on top to be a weight, that doesn't rescue it. So it's not as if it's because they're lighter, they grow faster. We think it really is something to do with the availability of carbon. And you can rescue that cotyledon removal non-sucrose mimic by actually adding sucrose back and lanolin to the tip. So it looks like it's that carbon source that's really important. Now, what we don't know is how this is being sensed. So, but there is something there that's changing this anisotropic ratio. So we can see this at a cellular level as well. So this is the 0% sucrose. You can see that increase in length of these. This is just looking at one cell in the base. Over time, you're seeing less growth in length. And also width, remember, you don't really see much of a wave in width in the normal condition, but you're seeing a little bit of a wave now here when you have this sucrose. So you're, all of a sudden, the width growth rate is getting regulated perhaps by that same acropetal wave. So there's an interesting shift there. Are there cell wall changes? And maybe they're related to pectin. That's probably why I'm here. So again, these can be really difficult to quantitate. So we're doing our best. But this is immuno for the LM20. Remember, that's the softer pectin, what we expect to be more fluid. And what we can see is that in the 3% sucrose case, we're starting to get more signal at these transverse walls than we ever had before. It's basically like an empty column in the 0%, and now we're getting some on these transverse walls. So the ratio between the two is getting closer to 1. So you're getting more soft pectin on that wall that's now elongating a bit more and giving you that shift to width growth over length. So you get more methylated homogalacturonan into the transverse walls. And the converse is also true with the demethylated pectin, although it's really a little bit less obvious. Quantitatively, we can see this, but the quantification's, again, a grain of salt. We're getting a shift away from those transverse walls. So you're getting less rigid pectin there, more soft pectin, which might relate to why you're getting that width growth that you don't normally get. So we can see an interesting effect when we look with the AFM. I forgot to change the title. That literally makes no sense, but we can call it figure 7. Okay. So if you do the AFM, again, these are the basal cells that are elongating. What you'll see is that these look more rigid than the 0% sucrose. You can also see they're a bit fatter than these, which makes sense given the measurements I've shown you. But it also looks like we're getting rid of that ratio, that difference between transverse and axial walls. And so here's just some quantification of that. So this is the 0% case where you see that difference, and there's less of a difference here, and also everybody's more rigid. So here's the ratio of the transverse to axial, which normally favors stiffer transverse walls, and here we've kind of gotten rid of that. Well, so this is kind of weird though, because now I'm showing you that the cell walls are stiffer, but we know they're still growing volumetrically the same. So if my walls are stiffer, how do I still end up growing the same amount, even though it's proportioned a little bit differently? And so that was puzzling to us. And so we went in and asked, well, is the pressure any different? And so we're estimating pressure here based on plasmolysis point. So it's not a perfect method, but it's what we have. And what you can see is that in 3% sucrose, the pressure is actually higher in cells at the base when they're growing. So the walls may be stiffer, but they've balanced that out somehow by regulating their pressure, so that the pressure to wall stiffness ratio is still facilitating the same amount of growth. And that's something, again, there's an interesting feedback there that we're not sure how it's happening. You can get that kind of dynamic actually out of a classical Lockhart model. And we're doing some modeling to look at that, but it's unclear what the actual mechanism might be in this case. So maybe you're interested in auxin because it's plants. So, and we know that changes in pectin can be affected by auxin. So we're looking a little bit at auxin transporters, and this is an example of PIN3. So these are auxin efflux carriers, and so they can change the direction of auxin flow depending on their polarity. And so in the 0% sucrose case, what we get is something that's sort of not very polar at all. It's sort of around the whole cell. But maybe here you can see it's a little bit stronger signal on the transverse walls. And we've done a bit of quantification here. So you have more signal on transverse walls than you do the axial elongating walls with sucrose, which means you end up with something like this. So you should have more PIN3 on these membranes, which will drive more flow across those walls. To get more auxin flow across those transverse walls. And we think that's facilitating their growth, perhaps through acid growth. And this is impaired in a PIN3 mutant, which is good. It's always nice to have mutants. So are there pH changes? We might expect that if there's more auxin flow across that transverse wall, that maybe we're going to see a drop in pH if you believe acid growth. And so we've been using this apofusion fluorescent protein system. And so what happens is you have RFP, which is pH insensitive, and GFP, which is pH sensitive. And you look at the ratio between the two, because they're both produced by the same construct and they go to the same apoplastic space in this case, and you say, what is the ratio between green and red? And that should tell you something about the pH of the system, because this will get lower when you have more acidic pH. And so our rough estimates, again, this is sort of preliminary, is that the pH ratio between the transverse and axial walls sort of is dropping in the 1.5% sucrose. And we sort of look at that as meaning that we're getting lower pH on those walls where more auxin is predicted to be flowing across. So we think there's some acid growth change that's giving this difference in width versus length. And this is impaired in proton pump mutants, but also if you add vancomycin, which is an inhibitor of these proton pumps. Okay, So the summary of this part goes like this. We think that there's this change in in polar transport of auxin that changes the pH. We think that's doing something, probably to PMEs and PMEIs on those transverse walls. We also have some hints that xyloglucan structure is changing a little bit on these walls as well, and it could be activating other enzymes like expansins, which we know are canonically activated by changes in pH. But the net result is we think these transverse walls in the sucrose condition are growing a little bit faster than they normally would, which gives you this slightly slower growing hypocotyl. It's getting a little bit fatter. But if you remember, I was kind of interested in whether it mattered that you were a little bit slower and a little bit fatter. Does anybody care? So we've designed some tests to look at this. And again, we're just starting to do this. And so we're looking at success of emergence. Because you could imagine just being faster, again, is awesome. Because you get out and you start photosynthesizing and you are the winner. But if you have different substrates, that might not be true. And these are not the best substrates, but it's what we had at the time. So we're using coarse sand and fine sand. So you have different particle sizes that you have to navigate through. But the thickness of the layer on top of the seeds on the plate is the same. And we orient the seeds so they're already ready to go up. They don't have to do any weird turning. And this is the sort of thing that we see. So when you have coarse sand, the skinny, fast one actually does a little bit better. But when you have fine sand, the fat, slow one does a little bit better. So the idea would be that it's not always advantageous to be fast. It's not always advantageous to be slow. But somehow if you can sense, sensing your environment and the carbon availability allows you to change this anisotropy, and you may be a little bit more successful than your friends. But it's obviously a pretty complicated matrix of interactions that we're just starting to explore. So I'm very quickly going to tell you a little bit about pectin gel mechanics, because I've been kind of going through and telling you, oh, well, pectin gel is really important, and if it's stiffer, that changes growth. But we want to know a little bit more about that as a material. So what does that actually mean? So how do you translate from this to the actual wall structure? And the way that we've been doing this is by really pulling stuff way out of the plant system and just making some synthetic materials and testing them. So first I'll show you some pure pectin gels. And we've developed another test with the AFM. So you will probably be familiar with creep tests. So these are classical tests that were done where you took like a section of hypocotyl usually, and you clamp it at one end and you throw a weight on the other end, and you just measure the deformation over time. So instead of these really fast elastic responses that I've been talking about all along, what we wanted to do is actually look at the way that the material flowed, because maybe that's a little bit closer to growth than an elastic band. So a good viscoelastic material to think about, which is essentially what we're testing here, is a memory foam mattress, which I got to sleep on last night. Thank you, Adrian. So when you get in it, you sink a little bit over time. So there's some time dependence in your deformation. And when you get off, at least when I get off, it takes a while to come back up, but it does come back up. So there's this time delay that might be a little bit more realistically related to growth. And so the classical creep experiment involves a weight on a whole organ, and we've simply miniaturized that using the AFM. So we push and hold a force and ask how the material relaxes away from that. So that's stress relaxation. You can push and hold the deformation, and there's a couple different ways to do it. But essentially, you're asking about that time deformation that happens. And then we take the data that we get and we put it through a couple of different really basic material models, but this one works pretty well. It's what we call a standard linear solid. You have elasticity from these springs, and you have the viscous flow from these dash pots, like a plunger and a syringe, and you can get parameters for these different things out of your models. So when we do this in pectin gels, what do we see? So what we have here are pectin gels that have different amounts of methylation. So this is 33% methylated. So a lot of that is available to calcium crosslink. And what we can see is that instantaneous stiffness, which is analogous to the indentation modulus earlier, is high. So the less methylation you have, the more calcium crosslinks you can make, the stiffer you come. That is good. That's what we would predict. It's also what's been predicted from food science rheology for a long time. This is not new. But what we have done is taken gels that have different percentages of methylation. So for example, here, half of it is methylated, and you can see it's a lot softer, and something in between. It's not a linear relationship. We've taken it another step further, and we've remade an average 41% demethylation by mixing 33 and 50. And we can see that it's not quite the same as when the average is actually 41. So you could imagine in a cell wall, it's not just that every single Homogalacturonan chain has exactly 50% methylation, but you might have some that have 70 and some that have 30. And what we find is that those synergistic combination of those things change the material properties as well. But what about the viscoelasticity? So what we can pull out of this is something called relaxation time. And I think what I'm going to show you is not the relaxation time, but I will tell you that what happens is that the gels are less stiff the more methylation you have, but they're also less viscous, so they're less thick. They flow better. And if it makes sense that growth, you might want your material to be more elastic, but also have a bit more flow in it. And so the way that we look at this, the easiest way to look at this from the models is actually to look at the ratio of elasticity to viscosity. And so because the elasticity changes, in order for that ratio to remain constant, you have to have the viscosity changing in the opposite manner. And so we end up knowing that as your pectin methylation goes down, so you have more available for crosslinks, you get stiffer and you flow less. Can we see that in a plant? So we go back to this case where we have more rigid transverse walls and more elastic axial walls, and we do these viscoelastic creep tests there too. And what we can see is that we do see a difference in elasticity from our model, but also in flow. So pectin chemistry doesn't just affect elasticity, it's also affecting the ability of the material to flow, which may be more related to growth, but we'll see. We still have to show that that is actually true. Okay, the last thing we don't have very much data on yet is we're starting to build more complex materials. And so to do this, we use cellulose from this bacteria, Gluconesidobacter xylinus, that makes cellulose outside its body. We take that cellulose and add in other cell wall components so that the architecture isn't going to be the same exactly as a cell wall, but hopefully we can get some information from it. And then we just test these big hunks of material for their material properties, and we can control them a bit better than we can, say, in a mutant in Arabidopsis. So here's a very brief example of some of the things that change. So if you have cellulose, cellulose plus pectin, cellulose and xyloglucan, you get differences in the weight, so the hydration, which makes sense. Pectin's a little bit wetter, can hold more water. You get changes in the depths of the material. So actually xyloglucan looks like it's really linking things more tightly. So proportionally of the same amount of cellulose in all these gels. It's just that you're getting a thinner material because of the xyloglucan. And we have some difference in Young's modulus as well, but we're just starting to explore this. And what we're hoping is by precisely controlling the components in the wall, we might get a better idea of what's actually happening in a plant cell wall. My last slide, going very long, is that we're starting to be able to do AFM-based mechanical measures on sections. So if you remember, everything I've showed you is from the epidermis, but all those tissue layers are connected to each other. And so you might want to know something about internal cells and their mechanics. And so what I'm showing you right now is the miscanthus stem cryo sections. They haven't gone through any fixation at all or embedding, except in OCT media. And right now we're just looking at vascular bundles because those are obviously going to be really mechanically different. That's a good place to start. But we're hoping to be able to look at primary cell walls as well that don't have these really rigid polymers too. But so you can see here, the phloem is softer than the sieve elements, which are softer than the xylem. That makes a lot of sense. And we can see some interesting differences between sieve elements and companion cells here within the phloem as well. And so this, we can relate this to some interesting galactan epitopes as well through work with Paul Knox, and that'll be coming out in Plant Phys pretty soon. But it just shows you that if you keep pushing the method, hopefully we can start asking even more questions and not just relying only on the epidermis. So hopefully you'll do that when you do AFM. Okay, most of this, pretty much all of this work that I've talked about today was done at the lab in Cambridge. But as Adrian moved, we've moved, as Adrian mentioned, we're now at UCLA. So we're continuing the work. These are the people who did it. 2 postdocs, Faraz Boudahir and Tom Tarode, and 2 PhD students, JJ Chen and Louis Kaplan. So material scientist and mathematician, while biochemist and cell biologist. It's a really interdisciplinary effort. And that's our money. Thanks very much.
transverse microscopy
Yes.
measurements of the modulus. You did it at constant deformation?
So the question is about the atomic force measurements and whether we're doing it at constant deformation. It's really difficult to do that with the way the machines work. So you actually have to do it with a constant force. So when we do the creep tests, we are choosing forces that give us the same deformation.
And how did you choose, in that case, how did you choose the force?
Yes. So in the plants, what we tend to do, and again, this is really hand wavy, we tend to say, well, we want an indentation depth that's no more than 5% of the depth of, say, the wall, because we want to make sure we're still in an elastic regime. But it's really hand wavy because most of the rules that have been used to model and determine these parameters from these experiments are with solid materials, and plant cell walls are cellular solids, and so there's some complicated geometry that goes into it. So generally we try and get, you know, either 5% of the depth of the wall or 5% of the depth of the cell, depending on the type of experiment. But one of the things we're doing now is sticking an AFM and a peeled epidermal layer on top of an inverted confocal and actually looking at what the deformation of the cell looks like under AFM. And it's not as beautiful and straightforward as a Hertzian model would want you to suggest.
One more question. If you change the force,
yeah,
and then do
yes,
everything at a different constant force, then you're checking a little more the longitudinal. The higher the force, the more the longitudinal component.
Yeah. So for the gels, we've done this a little bit more carefully. So because because it's a continuous media. And so you can look at, if you increase your indentation depth or force, you can look at the linearity of the response and things like this, and the gels look pretty good. I'm a little bit worried about doing those kinds of experiments right now in plants, just because what we think is happening is at one point you'll deform a wall, then you'll be deforming the whole wall, then you'll be deforming those walls and the cross walls and maybe the tissue underneath it. And so it's not as straightforward as you would expect based on simple material mechanics because you have that cellular architecture. But maybe we can talk about it a bit after too. Hi.
How does the hypocotyl node go up?
How does the hypocotyl node go up? So please correct me if I'm wrong. I believe it has to do with statoliths in the endodermis in the hypocotyl. So those gravity sensing starch containing granules that change probably auxin transport. Yeah, and then keep it growing up. Yeah. Hello.
Two observations. One that follows up on what Randy's question was getting at. And that is, I noticed that you had hotspots where the three anticlinal walls come together. So that makes perfect sense because you've got more solid material in that location.
Yeah.
And you have a Poisson effect, kind of a mechanical constraint.
Yeah.
Okay, so the question there is, how do you compensate for that extent? And the other, not question, but observation, is that you show that when you have more fine sand, the sucrose, which causes an expansion, they're more successful. Actually, that makes sense because if you look at the circumferential expansion, it propagates a fracture.
Okay, so it's better at pushing through those grains, which are more densely packed in the fine grain. Thank you. That's very helpful. I can go back to the first point, which is, and especially this has to do when you're doing sections as well, an AFM on sections. So you get these sort of nice trijunction middle lamellate areas where what everybody does right now, and whether or not that's a good thing to do, is they assume that you have an incompressible material. And so all these equations, you're using a Poisson ratio of 0.5. So you make a lot of assumptions. I think that, and definitely, so the wall, because that geometry becomes important as well, as you said, because you have a thicker amount of material, it may appear stiffer because it has less room to move in. We haven't started doing that yet, but you could, there's a lot of simulations you can do, simulated indentations in finite element models, which could be a way for controlling for that.
It's going to recommend
Yeah, I think that's probably what we'll do. So we'll just make a section and indent on it in silico. Yeah.
There's just one more question that I was interested in. I assumed, and correct me if I'm wrong, that when you're talking about the pectins, you're talking about the pectins within the cell wall.
Yes.
Question is, what is happening in the middle lamellae and the galactouronic acids that are binding the cells together, particularly where you have those three anticlinal walls?
Those junctions. So, yeah, I can repeat the question. So it's asking, so I'm making some conclusions about pectin in the cell wall, but I'm not making distinctions between where in the cell wall we're looking. So the middle lamella, which is responsible for the adhesion of cells together, is that different than, say, the proximal primary cell wall to the cell, which may be more growth regulatory. So our measurements right now haven't been fine enough to really tell us the difference between middle lamella and proximal, like right next to the cell. My postdoc Faraz is really interested in looking at cell adhesion and its maintenance. And so he started looking at some actin mutants that we can see have mechanical differences, but I'm hoping that this section technique, really fine tips, really fine scans, might actually let us see differences in middle lamella versus primary cell wall. I think that's probably the best way to go. In the Galactan paper that's coming out in Plant Phys, we can see that there's, in a single wall, in a sieve element, there's a gradient of binding of the antibody for galactan, and there's a gradient of mechanics across a wall. So we're starting to be able to see that kind of resolution, but we don't really have the answers yet. Hello.
I'm wondering, have we ever seen any effect on the plasma side or the number with the arch, the pectin methylation?
So the question is whether we've seen any change in plasmodesmata, number, size, maybe even opening with changes in pectin methylation. We haven't looked. So that is a bad answer for you, but it is the answer. So we haven't looked. One might expect that the more rigid the gel gets, maybe the less easy it is for plasmodesmata to dilate. I know that there are people looking at the callose deposition, and we've been doing some measurements for them on what callose does to the material properties of the wall. Because callose accumulation, maybe you know more, better than I do, callose accumulation around plasmodesmata can really restrict also their dilation. But it's a question that sometimes we get asked, and I don't have a really good answer for it. I mean, I think the only way to look would be to ask whether symplastic movement is different when you have the PME induction versus the PMEI. But we haven't done it yet.
In the interest of time, let's thank Siobhan.
Thank you for coming.
This has been a production of Cornell University, on the web at cornell.edu.